Can aging eggs be rejuvenated? Attention-grabbing headlines like this came up all over the place after a very early preclinical study was just presented for the first time at a conference in Scotland. Thankfully, one of the fellows in training where I work at RMA of New York did some sleuthing, and she was able to track down one of the authors on LinkedIn and got the preprint manuscript so that I can share with you the study behind the headlines, what they actually did and found, and why it matters. By now, we've all heard that the biological clock and its effect on egg quality can be a major driver behind infertility, miscarriage, and failed IVF cycles. The key reason behind it is something called chromosomal count errors. As we get older, the cellular machinery, think of it as like IKEA filing cabinets that are supposed to keep the chromosomes organized and neatly lined up for cellular division, starts to fall apart, and this leads to something called aneuploidy, where eggs end up with the wrong number of chromosomes, either too few or too many, and this results in embryos with the wrong amount of DNA. These embryos typically won't implant, so it's a cycle where you didn't get pregnant, or they may implant for a little bit and result in a first trimester miscarriage. The study in question focuses on key proteins that are involved. They're like the filing cabinet that keep the chromosomes organized during egg maturation and development. It's called Shugoshin-1 or SGO1, and in Japanese that literally means guardian spirit, which I think is very fitting because this protein plays a really protective role in egg cells, preventing premature separation of matching pairs of chromosomes that need to stay together until just at the right moment when the egg cells are supposed to undergo that genetic reorganization called meiosis. And this, generally speaking, happens at two time points: when the egg is maturing and ready to ovulate and when it undergoes fertilization. So in one of their experiments involving the mice model, they showed that this SGO1 protein helped preserve the glue called cohesin that keeps chromosome pairs together. And with age, we know that the amount of SGO1 in eggs drop, which can lead to this glue falling apart and failing, and this can allow chromosome pairs to split too early. So they demonstrated that by adding the SGO1 protein back into the mouse eggs, it reduced these errors dramatically. They were able to replicate this in human eggs that were donated for research, designated for discard in the lab, where they were able to inject the SGO1 into older human eggs, and it did cut chromosomal errors in about half. This is a scientific breakthrough. If we are able to correct the wear and tear of aging at the molecular level, we can protect the machinery that holds the chromosomes together. Maybe we can reduce the age-related fertility problems that we see and improve IVF success, or even reduce the risk of miscarriage in older women. As exciting as this sounds, I want to caution you that there are major limitations. This is really early stage research, and it's not really been done yet properly in humans. It's only been done on discarded eggs. There have never been embryos created or actual pregnancies. So no live births, no IVF outcomes have been studied using this SGO1 supplementation. And while it's a major part of the issue, ovarian aging is so much more than just having the wrong amount of DNA. Egg quality can be affected by mitochondrial dysfunction, DNA damage, and other forms of cellular wear and tear that this SGO1 protein can't necessarily fix. Not to mention, we lack a lot of technical details. We would need to know how to inject this protein into eggs just at the right time, in the right amount, and without damaging the egg. So there are going to be ethical and regulatory barriers. We need to make sure that there is extensive safety testing before any clinical use. And here's something else you should always pay close attention to. 3 of the authors on this paper co-founded Ovo Labs, which is a biotech company. They have equity in it. And it's really important that these types of findings are reproduced by other scientific groups that don't have stock in similar companies, because this is a form of bias. Now, we see this a lot in our fields. If you look at the early studies done on Receptiva test or the ERA test, a lot of them are authored by the people that are the inventors of the test that are selling it. This is exciting proof of concept research showing that molecular machinery maybe can be modulated even after aging sets in. And this opens up a fascinating line of investigation into how to address egg quality as we age. But for right now, this is science, not medicine. We're pretty far from being able to inject your eggs with this rejuvenation protein. And I would caution patients, because I don't actually love the term rejuvenation. It's been co-opted by wellness industries, supplement companies, and even clinics that are offering experimental, unproven approaches to restore egg quality. But the science being described here is not something that at this time can be replicated outside of a highly controlled research setting. But it does give me hope that one day we may have tools to help patients who are preserving their fertility or trying to get pregnant overcome age-related reproductive challenges.